Hey, Hammy here, coming at you with section 12.4, in which we now look at the first step of the central dogma of biology, in which we go from DNA to mRNA. So the first step in getting the information from the genes out and being made expressed as a protein. So first step is transcription. Okay, remember, transcription becomes before translation. So if we remember back to the last video, the central dogma of molecular biology says that the information, okay, is all stored in DNA, and then it's transcribed into mRNA, kind of a messenger that can leave the nucleus and is taken out to the ribosome and then translated into the final expression as a protein. Okay, so this first step right here, this first step, transcription, is where we need to go into a piece of the DNA that serves as the template for that gene. Okay, the DNA member is double-stranded, so it needs to be unzipped, and then on one part or one half of that DNA is where the gene would be. That provides the template to code for the mRNA. And that's just done using the C equals G and A equals, remember, instead of T now on the RNA, we will put a U. Okay, but if there's a T on the DNA, then that will make an A on the RNA. And when you get done, you get what's called a pre-mRNA transcript. That's going to go through some editing that we'll talk about in this video before it leaves the nucleus. And the sequence on that pre-RNA transcript is just a complement or a flip of the gene that is located on the DNA. It's important to understand in eukaryotes, a single chromosome is a very long molecule of DNA. Okay, it can be hundreds of thousands of base pairs long, and it can have hundreds or thousands of genes on them. Okay, so the genetic information in the gene will tell us, like reading a sentence, okay, it'll tell us the order of amino acids, okay, that will make up that protein. The amino acids, then remember, if you remember back to protein structure, the order of amino acids determine how they interact with each other to clump up to make that secondary, tertiary structures. The segment of, we don't have to unzip the whole chromosome, the whole strand of DNA, just the segment or piece of DNA that has the gene on, okay, is unzipped, okay, and we're going to use, instead of DNA polymerase, now we're going to use RNA polymerase. Is going to come in, read the genetic information on the DNA, and transcribe it or rewrite it into an mRNA. Remember, M stands for messenger RNA. Okay, so the completed transcript contains the information from the gene, but in a mirror or complementary format. Here's just a picture of that happening. So here's our DNA right here, kind of the aquamarine here color. It starts down here at a promoter, okay, a promoter region, which signals mRNA to come in and attach, the RNA polymerase to come in and attach. And notice it's just reading up this side over here. And where there's a C, it puts a G. Where there's a G, it puts a C. Where there's a T, it puts an A. Notice if there would be an A over here, like right here, it puts a U because it's RNA. Okay, so the RNA polymerase will read all the way up through. At the end of the gene, there's usually a termination sequence that causes it to fall off. Now notice, this is kind of a cool picture. This is what we call a lamp brush chromosome. Lamp brush chromosome. This happens especially in some lower eukaryotes in the early development stages where they need a lot of protein products. If you think of our DNA strand kind of being down the middle, these little loops, they cause these hairpin loops where there's just so much of these mRNA transcripts hanging off of them. So as this reads up through there, the next RNA polymerase is going to attach here and start reading before this one is even done. And you get several of them doing that, and it looks like kind of like a hairy brush. Okay, that's hence the term lamp brush, where you have all these mRNA transcripts hanging off of the chromosome because they're reading. They want a lot of mRNA transcripts because they need a lot of protein because they're growing so quickly. But kind of a cool phenomenon that we can observe underneath the microscope. Again, now notice that when it was done reading the gene, we called it a pre-mRNA transcript. Pre because before leaving the nucleus, it has to be processed. Okay, now how is it processed? Well, one of the first thing that's done is we add a cap and a tail. Okay, the cap is what we call a modified guanine cap. Okay, so remember guanine is a nucleic acid. Okay, a type of nucleotide that's sort of modified. And if you look, this cap will help kind of determine where the ribosome is going to attach and start reading once it gets out to the cytoplasm to start putting together the amino acids and make the protein. On the end, we add a poly-A tail. Poly, remember, means many. Adenines. Adenine, 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 up to 150 to 200 adenines, okay, go on the 3' end. This helps, number one, helps it get out of the nucleus, and number two, it inhibits degradation of the mRNA. So there's these hydraulic enzymes out in the cytoplasm that will start to break down on the ends, and if it gets into the gene, it's not going to make a functional protein. Okay, so the poly-A tails kind of think of something nipping at your heels. Okay, as these enzymes nip at the heels of this mRNA, it's just going to clip off some of these adenines. Okay, it's going to clip off some of these adenines, and so it takes it a while before it will degrade enough that it doesn't work anymore. Okay, so that poly-A tail is just kind of a buffer or protection when it gets out to the nucleus, or out of the nucleus, out into the cytoplasm. Also, before it leaves the nucleus, part of the mRNA processing is the removing of the introns. Okay, so you have segments of that gene that are needed for the protein and other segments that are not. Okay, the pieces or segments that are needed are called exons. I always remember exons are expressed. Okay, the introns we used to call junk DNA. They're the pieces of parts that are not needed. Okay, this says the introns are in between the exons. I always remember the introns are cut out. Okay, so the exons expressed, left there, put together. The introns cut out. Okay, and this allows the cell to pick and choose which exons go into a particular mRNA. And we'll talk about that a little bit, why, on the next slide. And this RNA splicing. Okay, so the primary transcript contains the introns and exons. And then the final transcript would just be the exons. And we'll see a picture of that here in the upcoming slides. How is this splicing done? In lower eukaryotes, the introns are removed by self-splicing. That is, the intron has the ability to kind of cut itself out of that pre-mRNA transcript. Higher eukaryotes, we use spliceosomes. Okay, just like splicing, when you splice two wires, you cut them and you fold them together and then you tape them up so you make a connection. Okay, a spliceosome is going to cut out the introns, and it's done by what we call SN RNAs, okay, small nuclear RNAs that are usually combined with proteins to make what we call a ribozyme. Okay, a ribozyme is an enzyme made of RNA rather than just protein. And these ribozymes come along and they cut out, kind of snip and cut between the exons. So they cut out the introns and then they connect or splice the exons back together. And so what you end up with is your mature mRNA transcript that can leave the nucleus and go out to the ribosome in the cytoplasm and be translated into that protein. Okay, here's kind of the picture of that happening. So here's our DNA. Here's the gene. Okay, this would be the gene that is red. Okay, so transcription happens. We get a pre-mRNA transcript that has exons and introns. Okay, so then the next step is to add our 5' guanine cap. And on the 3' end is our poly A tail. Okay, so it's transcribed. We add the 5' cap and the poly A tail. Okay, the next step then is the splicing. Okay, done by the spliceosome, these little green egg blobbies here. Okay, so there you see they're wrapping themselves here around the introns. The introns are removed. The exons are spliced back together. So our final transcript then is just the exons with a 5' cap and also the poly A tail. Okay, it can leave through the nuclear pore, go out into the cytoplasm, and that 5' cap helps find the ribosome and attach, where then translation can occur. What is the purpose of these introns? Why have junk DNA? Well, as this is a topic that's still being researched quite heavily, as the number of protein coding genes does not keep pace as the complexity of the organism increases. So the proportion of the genome that is introns also increases. Why is that? How does that increase complexity? Well, possible functions is that sometimes you will take the same gene and different parts may be exons or introns depending on what you want your final product to be. Okay, so it's kind of like changing the punctuation and changing the words a little bit in a sentence. If you cut out different words, the sentence will look very different and have very different meaning. So if we want one protein product, we would splice it and cut out certain parts. But if we want a slightly different protein product, okay, we might splice out different introns and put different exons together. So from one segment of DNA, okay, you could have multiple types of protein products that are made just by differential splicing. We think introns might regulate gene expression, and we'll get into that a little bit in the future chapter. And introns may encourage crossing over during meiosis. And again, this is something that is a lot of research is still happening. We don't understand. Okay, we used to call introns used to equal junk DNA. But now we're not so sure that's the proper term. Not necessarily junk. Sometimes they're exons, sometimes they're introns, depending on differential splicing. They may play roles that we don't yet understand. Okay, it might buffer for mutations. It might play a role in how and where crossing over happens. And so as this research continues, probably more and more will be added to the biology textbooks. We'll refer to them less and less as junk DNA, and they'll have more and more specific purposes. Okay, so what we end up with here is our final mRNA transcript, which has taken that code from the DNA and made a copy or a message on messenger RNA. It's going to leave the nucleus, and the next video we'll look at the final step, which is translation, taking that mRNA code and translating it into a protein.